Every breakthrough treatment you've ever heard of, from life-saving cancer drugs to groundbreaking vaccines, followed the same rigorous path. That path is the clinical trial phase system, a carefully designed progression that transforms promising laboratory discoveries into proven medical treatments. Today, we'll explore the four distinct phases that guide medical research, understanding what happens in each phase and why this systematic approach is essential for safe, effective healthcare. The clinical trial phase system is built on a simple but powerful principle: gradual escalation. Each phase asks increasingly complex questions while involving more participants and gathering more comprehensive data. This progression typically spans 8 to 15 years and costs hundreds of millions of dollars. But this investment ensures that by the time a treatment reaches patients, we have robust evidence of its safety and effectiveness. Only about 12% of treatments entering phase 1 ultimately gain approval. This high attrition rate reflects the system's effectiveness at identifying truly beneficial treatments while protecting patients from ineffective or harmful interventions. Phase 1 marks the critical transition from laboratory to human testing. These studies typically involve 20 to 30 participants and focus on one fundamental question: what is a safe dose? Primary objectives are safety assessment. Researchers carefully monitor participants for side effects, starting with doses much lower than those that caused problems in animal studies. They gradually increase the dose in small groups of participants, watching for any concerning reactions. Pharmacokinetics. Phase 1 studies track how the human body processes the treatment, how quickly it's absorbed, how it's distributed throughout the body, how it's broken down, and how it's eliminated. Biomarker analysis. Researchers often collect biological samples to understand how the treatment affects the body at a molecular level and to identify potential markers of response. Most phase 1 studies use a 3+3 design. 3 participants receive each dose level. If no serious side effects occur, the next group receives a higher dose. If serious side effects occur, additional participants may be added to that dose level before deciding whether to continue. Participants undergo intensive monitoring with frequent clinic visits, blood tests, and detailed assessments. This close observation helps researchers detect even subtle changes that might indicate safety concerns. Phase 1 participants often have advanced disease that hasn't responded to standard treatments. For these individuals, an experimental treatment may represent hope when conventional options have been exhausted. Participants must understand that phase 1 studies are primarily about safety, not treatment. While some participants may experience benefits, the main goal is determining safe dosing for future studies. Success criteria. Phase Y success means identifying the maximum tolerated dose, the highest dose that can be given without causing unacceptable side effects. Even if no participants are cured, establishing a safe dose range allows progression to efficacy testing. Phase 2 studies expand to 100 to 300 participants and shift focus from safety to efficacy. The central question becomes, does this treatment work for the intended condition? Primary objectives are efficacy assessment. Researchers measure whether the treatment produces meaningful clinical benefits. For cancer studies, this might mean tumor shrinkage. For heart disease, it could mean improved cardiac function or reduced hospitalizations. Activity determination. Phase 2. Studies determine whether the treatment shows enough promise to justify the substantial investment required for phase 3 testing. Biomarker development. Researchers identify biological markers that might predict which patients will respond to treatment, helping to refine patient selection for later studies. Study design variations consist of single-arm studies. Many phase 2 studies give the experimental treatment to all participants and compare results to historical data from similar patients who receive standard care. Randomized phase 2 studies. Some phase 2 studies randomly assign participants to receive either the experimental treatment or current standard care, providing a more direct comparison. adaptive designs. Modern Phase 2 studies may modify their approach based on accumulating data, such as focusing on patient subgroups showing the most benefit. Phase 2. Participants are more carefully selected than in Phase 1. Researchers often focus on specific disease stages, prior treatment histories, or biological characteristics to maximize the chance of detecting treatment effects. Many Phase 2 studies now incorporate precision medicine approaches, selecting participants based on specific genetic or molecular features that suggest they might respond to the treatment. Each study defines one main outcome measure that will determine success or failure, such as overall response rate or progression-free survival. Studies also examine additional outcomes like quality of life improvements, duration of response, and effects on biomarkers. Phase 2 studies must be large enough to reliably detect clinically meaningful treatment effects while avoiding false positive results. Phase 3 represents the definitive test of a new treatment. These large-scale studies, often involving hundreds to thousands of participants across multiple countries, provide the evidence needed for regulatory approval. Primary objectives are comparative effectiveness. Phase 3 studies directly compare the new treatment to the current standard of care, determining whether the new approach is superior, equivalent, or inferior. Clinical significance. Beyond statistical significance, Phase 3 studies determine whether treatment differences are large enough to matter to patients and clinicians. Comprehensive safety. The large participant numbers in Phase 3 studies help identify less common side effects that might not appear in smaller studies. Study design features consist of randomization. Most Phase 3 studies randomly assign participants to treatment groups, eliminating bias and ensuring fair comparisons between treatments. Blinding. When possible, neither participants nor researchers know which treatment is being received until the study ends, preventing expectations from influencing results. Multi-center design. Phase 3 studies typically involve many medical centers, ensuring results apply to diverse patient populations and healthcare settings. Statistical considerations are statistical power. Phase 3 studies are designed with sufficient statistical power to detect clinically meaningful differences between treatments, requiring careful calculation of sample sizes. Endpoint hierarchy. Studies define clear primary endpoints that will determine regulatory approval, along with secondary endpoints that provide additional valuable information. Interim analyses. Most Phase 3 studies include planned interim analyses where independent experts review accumulating data to ensure participant safety and study integrity. Regulatory considerations are regulatory input. Phase 3 study designs are typically discussed with regulatory agencies before implementation to ensure the study will generate data suitable for approval decisions. Quality standards. Phase 3 studies operate under strict quality standards, with regular monitoring and auditing to ensure data integrity and participant safety. Phase 3 participants are typically representative of the broader patient population who would receive the treatment if approved, providing evidence of effectiveness in real-world conditions. Phase 4. Studies begin after a treatment receives regulatory approval and enters routine clinical practice. These post-marketing studies monitor long-term safety and effectiveness in real-world conditions. Primary objectives are long-term safety. Phase 4 studies identify side effects that may only become apparent after years of use or in larger populations than tested in pre-approval studies. Real-world effectiveness. These studies determine how treatments perform in everyday clinical practice with diverse patient populations and varying treatment circumstances. Comparative effectiveness. Research. Phase 4 studies often compare approved treatments to each other, helping clinicians and patients understand which treatment works best for specific situations. Study types consist of registry studies. Large databases track outcomes in patients receiving specific treatments over extended periods, providing insights into long-term effectiveness and safety. Real-world evidence studies. Researchers analyze electronic health records and claims databases to understand how treatments perform in routine practice. Safety surveillance. Ongoing monitoring systems track adverse events reported by healthcare providers and patients, identifying potential safety signals. Regulatory requirements are mandatory studies. Regulatory agencies may require specific Phase 4 studies as a condition of approval, particularly for treatments approved under accelerated pathways. Safety reporting. Companies must continuously monitor and report safety information, updating product labeling as new information becomes available. Phase 4. Results influence treatment guidelines, helping clinicians understand optimal treatment selection and sequencing. These studies provide data on cost-effectiveness and health economic outcomes that inform healthcare policy and coverage decisions. While we discuss trial phases separately, they're interconnected parts of a continuous learning process. Each phase builds on the previous one while informing the next. Phase 1 to 2. Successful identification of a tolerable dose with an acceptable safety profile allows progression to efficacy testing. Phase 2 to 3. Evidence of clinical activity and a favorable risk-benefit profile justifies the substantial investment in Phase 3 testing. Phase 3 to approval. Meeting predefined efficacy endpoints with acceptable safety enables regulatory submission and potential approval. Modern drug development incorporates adaptive approaches that allow protocol modifications based on accumulating data, making the research process more efficient and informative. Biomarker research initiated in early phases may lead to companion diagnostics that help identify patients most likely to benefit from treatment. Traditional phase boundaries are evolving as researchers develop more efficient approaches to clinical testing. Improvements like seamless phase designs, Phase 1/2 studies. These seamless designs allow efficient progression from dose finding to efficacy assessment without the traditional pause between phases. Phase 2/3 studies. Adaptive designs may start as Phase 2 studies and seamlessly expand to Phase 3 based on interim results. Master protocol approaches like umbrella trials. These studies test multiple experimental treatments for a single disease, sharing infrastructure and control groups for efficiency. Basket trials. These trials test one treatment across multiple diseases that share common biological characteristics. Platform trials. Perpetual platforms. These ongoing studies allow new treatments to enter and leave the trial based on performance, creating a continuous research platform. Not all clinical trials succeed, and understanding what constitutes success or failure in each phase helps interpret research results appropriately. Success definitions by phase look like: Phase 1. Success: establishing a safe, tolerable dose regimen, even without evidence of efficacy. Phase 2. Success: demonstrating sufficient clinical activity to justify Phase 3 testing, based on predefined efficacy thresholds. Phase 3 success: meeting primary endpoint criteria with statistical significance and clinical meaningfulness. Phase 4. Success: confirming safety and effectiveness in real-world use, supporting continued clinical use. Plus, we learn from failed trials. Trials that don't meet their primary objectives still contribute valuable scientific knowledge, helping researchers understand disease mechanisms and refine future approaches. Regulatory considerations. Even successful Phase 3 trials must undergo rigorous regulatory review before treatments become available to patients, ensuring comprehensive evaluation of benefits and risks. The clinical trial phase system represents one of modern medicine's greatest achievements, a systematic approach to developing safe, effective treatments while protecting patient welfare. Each phase serves an essential purpose. Phase 1 establishes safety. Phase 2 demonstrates activity. Phase 3 proves effectiveness. And Phase 4 ensures real-world performance. Together, they create the robust evidence base that modern healthcare requires. This systematic approach has given us countless life-saving treatments, from antibiotics and vaccines to targeted cancer therapies and gene treatments. While the process is lengthy and expensive, it ensures that patients receive treatments backed by solid scientific evidence. As clinical research continues evolving with new technologies and methodologies, the fundamental principles remain constant: systematic evaluation, patient safety, and scientific rigor. Understanding these phases helps everyone involved in healthcare, from researchers designing studies to clinicians treating patients to individuals considering trial participation. Each phase represents a critical step toward the ultimate goal: improving human health through rigorous scientific research.